Okay, it's 1+1 now, so we're going to make a start. Good afternoon, everyone, and welcome to this session on transitioning from academia to industry in antimicrobial resistance, which is brought to you by Microbiology Society as part of the Knocking Out AMR project. My name is Magdalena Karnikowska, and I'm the Chief Executive Officer at Cyticom. Today, I'll be speaking alongside Dr. Soumya Palil, who is the co-founder and CEO of Bridget Bio, as well as Dr. James Priest, Chief Operations Officer at Biofis. In today's session, we will each share how we made that jump from research to real-world application, what it really takes to move a promising technology out of the lab into a suitable company. First of all, each one of us will give a 12 to 15-minute presentation, starting off with Soumya, who recently spun out an antifungal therapeutics company from the University of Aberdeen, followed by myself, talking about my journey from an academic spin-out with a clever idea, but not a clear market fit, to a focused business now developing rapid antimicrobial susceptibility testing diagnostic for sepsis. And we will end with James Priest, who specializes in actually helping early stage companies, not only diagnostics, to really find that right product market fit, making them commercial. We will then move into our Q&A session. So please do send any questions that you might have throughout the talk into the chat box or the Q&A box. So without further ado, over to you, Soumya.
Thank you, Magda. I'm just going to share my slides here. Okay, can you see that? Okay. Right, okay. Thanks, Magda, and good afternoon, everyone. It's a real pleasure to be here today and part of this webinar, which is organized by the Microbiology Society. As Magda said, my name is Soumya Palil. I am the CEO and co-founder of Bridget Bio. So I get asked this question a lot: what motivated me to transition from academia to industry and set up my own business? So in today's talk, I will touch on this and also highlight some of the entrepreneurship training and funding opportunities that are specifically available for academics who are also thinking along the lines of commercialization of their research ideas. We all strive to achieve our research to get to far-reaching societal and economic impact. And for me, the main reason to kind of transition into a product developer was to create that much greater real-world impact through novel solutions to global challenge. And for us to transform the kind of cutting-edge scientific inventions into solutions, we need to embrace that entrepreneurial mindset and foster skills that bridge the gap between creativity and real-world outputs. And this was my reasoning behind thinking about a spin-out company and finally allowing me to become a co-founder and a CEO of Bridget Bio. So I'll start with a brief background of my career journey so far. So I came to the UK, funded by the British Council Commonwealth Scholarship to do my master's in biotechnology at the University of Aberdeen, which was followed by a PhD in molecular biology, which was funded by Knowledge Transfer Partnership between the University of Aberdeen and Pfizer. So some of you might be familiar with Knowledge Transfer Partnerships, or KTPs. This is where it's a three-way partnership between a company and a university, and there will be a KTP associate who works on the project and bridge between industry and academia. So I was a KTP associate on that project, and this gave me the opportunity to gain first-hand experience of how innovation is driven through industry academic partnerships. This KTP program won the Best Partnership Award in Scotland in 2011. And this also allowed me to graduate from my PhD as a qualified antibody engineer. So my PhD project was developing monoclonal antibodies to break communication or quorum sensing in gram-negative bacteria, specifically Pseudomonas aeruginosa. So I had this idea of developing these antibodies from my PhD into novel therapeutics to treat gram-negative, drug-resistant gram-negative infections. I was fortunate enough to receive the Royal Society of Edinburgh Commercialization Fellowship to develop that idea. This prestigious program supports academic entrepreneurship and accelerates innovative science and technology into new businesses in Scotland. Although so that was a great program which gives you tailored business training, mentorship, and funding, business funding to the company. I was not able to make that business idea into reality at that stage, and this was because at that time an anti-infectives company developing biologics therapies like antibody-based therapies was not of interest for the funders. So we're talking about in 2011, and this at that stage AMR was not widely recognized as a global challenge, and this was before the WHO's first global report on AMR surveillance was published, and of course the landmark report on antimicrobial resistance by the economist Jim O'Neill. So things were different at that time. So although I was not able to set up a company, the commercial network that I built and the trainings and mentorship that I received allowed me to take up the role of the head of Scottish Biologics facility at the University of Aberdeen. The Scottish Biologics facility is a recombinant antibody discovery center, and there I initiated, managed, and delivered a portfolio of antibody discovery programs, including the 4 million Alzheimer's program, which was funded by the industry partner TowerX Therapeutics. So along with that, I also have a position as a senior research fellow at the University of Aberdeen. So I have my research activities, but also do teaching, which include biotechnology, antibody technologies, and also biobusiness, which I teach both in Aberdeen and at the University of Glasgow. I'm also the enterprise and innovation champion for the school within our university. And my role here is to promote and signpost fellow academics who are interested in commercialization and industry partnerships. So all projects that we take on in the Scottish Biologics facility have a focus on real-world outcomes and global impact. So collaborating with Professor expert at the University of Aberdeen, we developed human monoclonal antibodies to novel drug targets that Professor Munro has identified through her extensive research on fungal cell wall remodeling. So these drug targets are specific to fungi, and they play a crucial role in infection and drug resistance in fungal pathogens. So this is the scientific foundation for Bridget Bio, which is a spin-out company, a recently new spin-out company from the University of Aberdeen. And in Bridget Bio, we are pioneering the development of next-generation immunotherapies to treat life-threatening, serious invasive fungal infections. So these invasive fungal infections are life-threatening in immunocompromised individuals, including patients who are undergoing chemotherapy and also other complex treatments such as transplantation and abdominal surgeries. Globally, 3 million people die each year due to invasive fungal infections caused by Candida and Aspergillus species. If you look at European ICUs alone, 79 of invasive fungal infections are diagnosed daily, out of which 29 patients sadly die in the next 30-day period. So there is a desperate global need for novel therapies against invasive fungal infections. The limitations of current antifungal treatments include high mortality rate, 40 to 80%, despite receiving treatment in several cases. Also, the the rise of antifungal resistance. So AMR is not just resistance in bacterial pathogens alone now. Antifungal resistance, or AFR, is on the rise. There are dangerous drug-drug interactions when administering current antifungals with people receiving chemotherapy. And some of these antifungals are inherently toxic. And we only have 3 classes of antifungals currently to treat patients. Dosing frequency is another challenge. So the most effective current antifungal drug is an IV formulation. So it needs to be administered daily, which results in prolonged hospital stay. So our solution is a novel immunotherapy, which can boost the immune system of patients and clear infection. So our antibodies are first-in-class, fully human, with a novel mechanism of action. And these antibodies have proven efficacy in infection models, which are clinically predictive of infection in patients. Monoclonal antibodies have increased safety profile, which means these drugs can be administered either as a prophylaxis or treatment along with chemotherapy. And the increased biological half-life of monoclonal antibodies means we can administer these drugs once a month as opposed to daily or weekly dosing with the current antifungals. We have also shown that these antibodies are effective against drug-resistant fungal pathogens of clinical significance. These include Candida auris, Candida glabrata, and Candida parapsilosis. So the early stages of our technology development, antibody generation, and the characterization were all funded by research grants, both into the Scottish Biologics Facility and Professor Carol Munro's research group. We filed the patent which described the generation of these monoclonal antibodies in 2021, and a publication followed. Then, in identifying the commercial potential of this project, we spoke to an organization called the Scottish Enterprise, which is the economic development agency of Scottish government based in Scotland. So Scottish Enterprise has a high growth spin-out program, which funded our early stages of commercialization. Both myself and Carol, we attended several business trainings, product development workshops, and also won business awards like the Converge Challenge, which is for Scottish universities, so the business ideas from Scottish universities, and also Scottish Regional Edge. So these competitions and these awards allowed us to raise the profile of the company and also build further partnerships. I was also selected to be part of a global business innovation program, which is funded by Innovate UK. These are called GBIPs, or Global Business Innovation Programs. This particular GBIP was focused in AMR, and this was in Switzerland. So it was an incredible opportunity, I have to say, to go and meet with funders, industry experts, and biotech pharma companies based in Switzerland. I was part of a cohort of 15 founders or CEOs from all over the UK. All these businesses were working on the AMR space. And then earlier this year, we were funded the Innovate UK iCure funding, which enabled the spin out of Bridget Bio. We spun out in July this year, and this funding was matched by Scottish Enterprise convertible loan note. And we also got funded the Stage 2 award by an organization called INCATE. INCATE is also based in Basel, Switzerland, and it's the European incubator for antimicrobial therapies. So companies like us at early stage and just coming out of academic institutions are allowed to apply for this funding from INCATE. So it comes with the Stage 2 funding comes with 250,000 euros of non-dilutive funding, but along with that, access to the industry experts within the INCATE network. So it's an incredible opportunity, I have to say, so which we can already start seeing the benefit of being a Stage 2 company of INCATE. So this is that journey which allowed us to kind of take a research program and convert or kind of transition that into a spin-out business. And I have to say that it has been an incredible learning experience for me, and I made sure that every opportunity was taken up, but it was also challenging at sometimes, but I have to say in general this has been a great experience for me. So I've put some challenges and opportunities here. I won't go through all of them, but I'll highlight a couple of them. The first one we all face is that access to funding, especially when you are at an early stage. So what we did was, we applied for several of these commercialization grants. These are all non-dilutive funding, so you're not really giving up any of your equity, the business equity at this stage. These are, again, great programs, I would say, because this kind of de-risks the business, because this has been, these applications are reviewed by a panel of industry experts, so the feedback that you get is really invaluable, and also it kind of validates your business idea. Building credibility is also challenging when you are setting up your first business. In academia, if you have a good publication record and with your conference presentations and your grant funding track record, you kind of demonstrate your credibility. But someone like me venturing into business for the first time with no proven track record, this has been hard. But the best we can do is to build a credible team around you, and also kind of build strong partnerships. I would also want to say the role of mentors and champions are very important, and also building that extensive industry network, which will increase the credibility and authenticity. And in my experience, winning awards and then taking part in business programs like GBIP also increases the profile of the company, but also the profile of you as a founder or a CEO of the business. So village to build a company, and I am ever grateful to our funders, ecosystem developers, university tech transfer team, advisors, mentors, industry experts, all for making Bridget Bio a reality. So I'll stop here and very happy to take any questions at the end, or if you want to hear more about the details of some of the programs that I mentioned in the talk, happy to talk more about it. So thank you.
Thank you very much, Soumya. I'm gonna take over the screen now. Can you let me know if that's sharing okay?
That's good, Magda. Yeah.
Thanks. Perfect. Well, thank you for this introduction, Soumya, and it's a very exciting journey and only a beginning of one as well. I I had a little bit of a different path to yours. So as I've already said, my name is Magdalena Karabikowska, and I am actually a scientist by training. I did my PhD in microbiology, and now I am a CEO of Cyticom. And my path here wasn't exactly planned. So I initially trained as a microbiologist. I also worked in clinical trials, and I worked in diagnostics in the reference labs of the UK Health Security Agency in Colindale. But I always really had an interest in translation. I was very curious about how discoveries that we see coming out of laboratories, academic or public health, actually applied in solving real-world problems. And in my efforts to understand that, throughout my PhD, I was very active in getting engaged with several strategy consulting projects across different industries, really. They ranged through automotive, educational technologies, but also healthcare. And that's really where I got a very first flavor of entrepreneurship and what it takes to actually take an idea outside of university into something that's commercial. I then got my really first startup experience through this BBSRC funded flexible talent mobility account placement that I did with Cyticom that I now run as the CEO. So Cyticom spun out of University of Warwick. I got to know the team really through proximity because I did my PhD in the lab next door. And having completed my PhD, I joined the company on this placement program, fellowship program, to work as an application scientist to really test the application of the company's technology into rapid detection of mycobacteria, because that's what my background was. I did my PhD in slow-growing mycobacteria. Throughout this placement, I really split my time between the lab as well as looking for the product market fit. This was done largely at the time via desktop research, as well as online meetings on Teams, on Zoom, because it was really throughout the COVID pandemic that it was happening. And at this time, I really didn't think that that's what my career was going to take me. I already had planned to move on to Colindale to train as a clinical scientist in microbiology. And I did that soon after. But about a year and a half into my training, the company called me and they've asked me whether I would like to come back and help them, this time in the capacity of the CEO. So I quit my training and that's what I did. And I really enjoyed putting this presentation together because it took me back 3 years to when I first joined Cyticom. And I'm going to show you a mix of slides that I was presenting back when I first joined and then how we grew as a business since then. So what you're seeing at the moment is one of the first presentations that I ever gave about 1 month into my role at the company. And at this point, the science was really brilliant. We had patented technology. It was very exciting. However, the story around that science was somewhat missing. We were marketing this shockingly fast bacterial detection device that could count living bacteria in 10 minutes. And we framed the problem that we were going to solve as a slow and outdated bacterial culture process, which all of you here will be familiar with, still reliant on the Petri dishes invented over a century ago. It was technically quite accurate, but commercially, pretty much it was quite useless. And the reason I say that is because whilst it identified the technological bottleneck, i.e. slow culture, which can take up to 48 hours, in the case of Mycobacterium, even longer, it showed the scientific opportunity for innovation here. We can speed it up, and that's what Cyticom indeed could do and can still do. But what it didn't do very well was, why do people care about the slow culture? And what is the pain point of that culture? Who is affected by it? So in that slide, we really skipped all of that. And therefore, when we were presenting this to investors and customers, they didn't really see any urgency of it. And if there is no urgency, that means that there isn't really any business case. So sort of the lessons from academia moving into industry, what we often do in academia is the problem that we're looking at is a technological challenge that it's worth solving. However, in industry, the problem must really link this pain point, the stakeholder, and a consequence. Now, at the time, we had a half-developed prototype, and we were marketing it as a universal detector. So it was quite, like I said, it was quite scientifically exciting, but commercially quite confused. We were looking at cosmetics one day and then fuel tanks the next. And what that really showed to the market, to customers, to investors, was that there was a clever bacterial detection gadget, which was exciting, but no customer, no defined market, and to them, there was no clear reason to exist beyond the science. We were really the classic example of a technology in a search of a problem. And this is very commonly seen in university spin-outs. So I quickly learned that when your market is everyone, your customer is really no one. Without having that defined customer pain point, our innovation lacked traction. And through that, I've realized that my first role, my first task was not to sell, but actually to listen. So I began my role really by going out there and conducting structured interviews, going to potential customer sites, understanding what the problems are, seeing them and experiencing them firsthand to really learn about it, and then understanding how we can adapt our current prototype to provide a solution to an actual commercial problem. I wanted to mention here as well that my first day on the job was during the Microbiology Society's annual conference back in Belfast in 2022. And through that, we actually had a great opportunity to speak to a lot of different people, mostly researchers, but also some industry experts from different market niches. One important bit that we did was to start collecting the names and the contact details for all of these people. We generated about 80 leads, and we then lined up 10 demonstrations of our technology at the time. And that really led to these customer interviews and the site visits. That was very, very helpful. We then mapped all of these different markets, and we did quite a simple calculation. We looked at the market size, the unmet need, and then also the customer's willingness to pay and at what price point. And based on that, we ended up focusing on and understanding that healthcare had a very great impact potential. However, it also had a lot of regulatory hurdles, but this is what we set on. So from this unfocused problem statement and without having a defined customer, we changed our problem statement into what we have now, which is the fact that clinicians that treat sepsis have to wait about 48 hours, if not longer, to receive results in terms of antimicrobial susceptibility testing. Now, this is really not good enough for the patient, because at the moment, a septic patient, for every 1 hour that the correct antibiotic treatment is delayed, there is a high risk of mortality. With the antimicrobial resistance on rise, as you all know, the likelihood of having putting the patient on incorrect antibiotic treatment is higher and higher. In fact, about 40% of the patients nowadays who are septic are actually associated with infection that's resistant to antimicrobials. And what we show at Cyticom is that rapid phenotypic testing could guide this therapy the same day rather than waiting for a couple of days. And through that, actually save times, reduce the number of days that patients spend in intensive care units, and help doctors make this smarter, data-driven decision-making. The technology was also an interesting lesson. When we first spun out and when I first joined the company, we were really talking about it in a lot of depth and detail, very much like a scientist. What we've learned was that it's much better to keep it simple because a lot of the potential audience that we were talking to was very quickly lost. So at the moment, we're presenting our technology as novel patented optical electrophysiology, which combines time-lapse fluorescence microscopy, bioelectricity, and data analysis to quickly deliver precise measurements of bacterial responses to antibiotics. And briefly, the way the process works is that we take a clinical sample, such as blood or urine, we expose it to a range of different antimicrobial agents, and we zap the sample with an electric shock. We then observe the bacteria that contaminate that sample under the microscope. If the bacteria are resistant, meaning that they survive the antibiotic treatment, they will quickly light up in response to that electric shock. On the contrary, susceptible cells will stay dim. And this is a significant improvement on the current standard of care whereby we have to culture the bacteria and wait a couple of days to get the same result to help the patient that's suffering a bacterial infection. So the key lessons from this was that in academia, we often trained to describe problems in terms of what we don't know. But in industry, what we don't know is important. However, what's really important for a problem is to understand what people cannot afford to ignore. And one book that I would like to recommend that really helped me at the beginning of this customer discovery is called The MAM Test. And there is a quote here from Rob Fitzpatrick, who wrote the book. If you talk about your idea, people will lie to you. If you talk about their lives, they will tell you the truth. And really what that's saying is, don't go to people with your solution. Go to people and understand what it is that is stopping them from doing the most efficient and effective job that they can, and understand what the problems are. Then tailor your solution to that problem and go back to them and offer that. And then develop a co-development partnerships where together you can come up with a product that people will actually pay for. So next challenge and next interesting kind of journey for me was how we think about a business model. In academia, again, we learn to describe value of our work in terms of knowledge gained. So Samia already touched on it. The kind of credibility comes from publications, citations, and new understandings. However, in industry, we must really describe the value of our technology and the proposition in terms of the problem solved, money saved, or money made. And the success is measured by the adoption of your product, revenue, and measurable outcomes. And that's really quite a big difference. So in our case, rather than talking about speeding up a cumbersome bacterial culture, we're now talking about delivering better patient outcomes, lowering the hospital costs, and producing a sustainable revenue stream. One of the things to get there that was quite key for us was to understand how the reimbursement works in sepsis care, and to understand really how our diagnostic tests will be paid for. So in the US, here in this slide, what you can see is it's to really how it happens in the US. So in the US, sepsis is actually a loss leader for hospitals. What I mean by that is that an average cost of treating a septic patient is about £25,000. However, the hospital gets a reimbursement of a flat fee of about £17,000, meaning that they lose on average £8,000 per every patient. Now, with a rapid antimicrobial susceptibility test like ours, we can actually flip this health economics. And because we can provide bedtime savings. When you think about an ICU patient, this bedtime savings are actually quite significant. If we could save the length of stay by 2 to 3 days, we could actually turn this loss-making reimbursement code into break-even or even positive. For a hospital of 500 patient beds, that's actually a $4 million of annual swing, $4 million of annual swing. They're looking also in the US at this increasingly more common model of value-based healthcare. What's quite important is also the risk reduction in terms of lower readmission rates that are associated with better patient outcomes if they're treated with the right antibiotic much quicker than currently possible. The team is also quite important. Again, Soumya already mentioned that, so I'm not going to go into too much detail, but really what you have to do as you're growing a business is to surround yourself with people that are much smarter than you in finance, legal, regulatory, and other expertise areas that you might not have had that experience in. And the role of these advisors and consultants and the team overall is to de-risk your decision making. The way I like to phrase it is the role is to uncover for you the unknown unknowns and making them either known unknowns or unknown factors. And that can really help you accelerate your business development, your go-to-market strategy, and many other challenges that you will come across as you run the business. So what did that journey give us? Well, transitioning from this unfocused, shockingly fast bacterial detector into a rapid antimicrobial susceptibility diagnostic test meant that we could unlock our credibility. Really once we've aligned our technology with the patient need, the economics, the validation, and the funding followed. So soon after, we managed to secure the Product Development Award from NIHR. We also secured a partnership with the NHS in Leicester. And at the moment, we're undergoing preclinical evaluation of our prototype to validate it as a proper diagnostic device and to get us ready for the regulatory approvals. A little bit about the project. The overall plan is to speed up the precise diagnosis and treatment of serious bacterial infections in blood. And we've got 4 objectives. Firstly, we're developing proprietary protocols for the testing using our patented bioelectricity method. We also putting together novel algorithms for drug susceptibility profiling using our data. And throughout the project, we're also working with health economists to create the value proposition and health economic modeling to help our commercialization strategy. And just now, we're actually also starting the patient and public involvement and engagement in preparation for clinical testing that we will have to do in the future that will help us inform the public why clinical consent for this kind of research is really important. And we hope that will then streamline patient recruitment in the years coming ahead. Broadly speaking, when you develop a diagnostic product, you have to validate it. We start off with testing our samples in lab media. We then move on to animal blood, and then the third step will be testing our approach in human blood. Regulatory strategy, as I've mentioned, is very important. We can touch onto this during our discussion, but that was a huge learning curve and very much supported by NIHR itself. And also understanding that understanding the go-to-market strategy and what it takes to develop your product, but then also commercialize it, and having that overview of how many years and how much money it will take you to get to the market from today is really the key in scaling your business. So in terms of some of the take-home advice for all of you listening here, I would say validate your customers as early as possible, even before you have a prototype, let alone finalize it. There are a number of programs out there that can support you in that journey, like the Innovate UK iCure, but also a lot of different early stage accelerator programs for entrepreneurs. I would also recommend taking a short executive courses. An example of a good one is Enterprise Tech run by the Judge Business School in Cambridge. And throughout all of this, don't be too attached to your original idea. Be ready to pivot the narrative, but stay true to your mission. So thank you, and over to you, JP.
Great, thank you, Magda. Let me know when you all can see my screen. I've just sent the request. Stuff. So there we go. That should be on the screen now for everybody.
I can't see it.
Okay. You may have to stop sharing your screen, Magda. I can still see yours, but I think I can.
Sorry. Yeah, it's shared.
Perfect. Well, thanks, Magda. And hi, everybody. And thank you all for the opportunity to speak to you all today. Like the 2 fantastic talks before me, I think what I'm hoping to do is just reinforce some of those excellent points made. But hopefully this sort of arms you with some more tips and tricks to sort of send you confidently off into the big scary world, of industry. So my name is James Priest. I'm the Chief Operations Officer at Biofizz, and we're a life sciences consultancy based in South Wales. And our goal is really simple. It's just to use our knowledge and our expertise to help bring new scientific ideas to life. So what I hope you're going to get out of this talk is just some first-hand insight of my journey in industry and how you can apply those to your current ambitions. So my scientific background is in molecular biology, where I originally spent several years working as a research scientist, and we were developing novel antimicrobials and antivirulent strategies. And that was before I took that proverbial turn to the dark side and started to venture into the world of product management and business development. And that was where I was a product manager at the company called Fluoretic, and we were developing rapid point-of-care tests for the detection of urinary tract infections. Now, similarly to these both, when I reflect on my career, I've almost worked exclusively within small business and startup environments. And this is where I've really gained an appreciation for the alchemy of new ideas, turning that nothing into something, creating something that's new and exciting, but importantly, translating those ideas into real world products. And that's something I get to do every day at Biofizz. I'm sure there are very easier ways to make money, but I also enjoy talking about the possibilities and bringing those what if ideas onto the market. So over the years, we've worked with a wide range of university spinouts, SMEs, international corporations, and we've supported projects across a vast number of sectors, including IVDs, medical devices, drug development, vaccines, food and drink projects all over the world. But the core offering of our business has always been consultancy, and we've been able to play key roles in teams who've achieved getting their first in vitro diagnostics onto the market, obtaining orphan drug designations, and also building quite large infrastructure aquaculture projects. Quite recently, we've begun to diversify our business offering, and we now offer a complementary laboratory CRO service. We've got a containment level 2 facility suitable for micro, cell, and molecular biology, as well as a confocal microscopy suite to offer, that real hand-in-hand offering for new clients. So some of the recent examples of the projects that we've supported include those sort of translation of ideas from academia into industry. And these are all very specific to the AMR community. So Microbia, these were associated with the University of Edinburgh. They developed an ATR FTIR technology that uses a biochemical fingerprint to rapidly identify bacterial species. And we were really useful in helping them redefine their commercial strategy and also conducted a third-party evaluation of their diagnostic. Disperazole, a spin-out of the University of Copenhagen, and they developed a novel cyclic di-GMP inhibitor, which forces the environmental trigger from biofilm to planktonic cell states. And we helped them strengthen their IP strategy with a composition of matter patent through the development of a novel scaffold with a new molecular library. And finally, QED were a spin-out of the University of Lancaster. They were developing rapid LAMP-based PCR technologies for food hygiene and fish stock health. We're now part of the management team brought in to bring their first products to market, setting up their first trials with customers and helping them convert that initial interest into revenue. So I hope that kind of sets the scene for you. As the consultancy, you have an opportunity to work with a wide range of ideas from academic founders. The diversity is brilliant. We're constantly evolving our approach, and we can take learnings from both past projects and then just apply them to new ones. So we can make sure we can avoid many of the common pitfalls when starting a business. And we also work with lots of clients who may have opportunity, to discuss or join their skill set. So beyond recommending that you talk to us, I wanted to make sure that you all had a benefit from attending today's session, and it's all about reinforcing some of the points that my co-presenters have mentioned. But this is 5 hard-learned lessons to help smooth your transition from academia to industry. Now, the assumption I've made is pretty general, but it assumes that you're all in the big wide world of academia, and you've probably heard lots of secondhand stories from those who've made this proverbial leap. Some might have been successful, some might have started their own business, joined a startup, and I'm almost certain that you've heard some horror stories from them as well. But hopefully I can share some guidance so that your journey is enjoyable and bears some sweet fruit. So point number 1 is finding the right opportunities. As Magda has highlighted, I think a university sometimes can be quite technology orientated, focusing on the cool, idea rather than the right problems to solve. It's that characteristic, well, my technology is faster, it's cheaper, and it's my favorite color. But the question I think that people forget to ask is, does the customer or the sector that you're looking to impact care? Your solution may be cheaper, but does it completely disrupt a workflow? That's almost vitally important to the NHS. Your solution is fast, but the existing solution may be fast enough. Does that mean I have to sit next to my machine for 10 minutes, whereas previously I could have walked away for 24 hours? So I think the lesson I'm trying to emphasize here is that industry or your mindset needs to always prioritize the customer. Now, don't get me wrong, they are tricky. They'll rarely tell you what you want. So as Magda has done very well, I think you need to look between the feedback and try and find what the real pain point or what the real opportunity is. So lesson number one is really about finding the right opportunity and understand what moves the needle for your customers. I think point number two, again, worth bringing up, is that during this journey, you're always going to want what the opposite side has, and I mean academia versus industry. A good example of this is around funding. There are lots and lots of academic funding opportunities that, at a university, you have access to, but you can work collaboratively with industry for those ideas that focus on fundamental science. But small businesses, unfortunately, are reliant on a much smaller and I think hyper competitive at this point in time pool of grants. So their research programs need to be really sort of focused. But as is done with many sort of stalwarts of the industry, these businesses regularly reinvest into R&D when they're successful. So, you need to focus initially, but that's not to stop you from creating multiple product lines for multiple applications. But you do need to focus on that niche market opportunity to start with. I think job security is a very poignant point to make at this point in time. Everybody is looking for that long-term security because they may have families to provide for, mortgages to pay for. There's also the added pressure that universities are typically shrinking their budgets at this point in time. But I think it's just worth highlighting that whilst you might be in a slightly more insular position within a startup, it's obviously highly funding dependent. Your runway is, cash is king. You're going to have to very cleverly manage your cash flow to do the activities that you want to do. And it might be a little bit more constrained than what you're used to in the university world. So I think just bear that in mind when it comes to looking at the other side. And I think finally, just to touch on IP and intellectual freedom, there's a big challenge to the publishing model. Everything that you've been working towards, you're going to have to keep secret, at least for a period of time until it's patented or it's protected with a design right or copyright or a trademark or something along those lines. Again, it doesn't mean that you can't publish and still use that as an important way to leverage interest in your products. But again, putting the cart before the horse, making sure that your idea remains valuable when you put all of this time and effort in. So I think, point number two, just bear in mind that the grass isn't always going to be greener, and certainly it's not going to change unless you nurture the opportunities that you currently have. So point number three, and I think it's probably the most important one I'd like to bring up, is that you have to have this change in mindset from being paper focused to being product focused. So point number three is very much product, but not perfect. I can't stress how important it is to create something tangible. The moment that you have a prototype or a working model or something physical, something tangible, that is when you're going to have the biggest change in your understanding of what this product needs to be, but also your biggest engagement with your customers. To oversimplify it, your research paper has worked once under ideal conditions. It's flawless because it's been imagined. I think the problem is when you ask people to imagine things repetitively, they may start imagining different things. So the point I'm trying to emphasize here is that on your journey, I want you to make something. It's going to be ugly, it's going to be oversized, it's going to be clunky, but I promise you that's where you're going to get the biggest developmental leap. It can be that preciousness, you don't want people to criticize your idea, but customers can be cruel, but I think they bring perspective, and they're always going to find a way to make your products better. So point number three is don't be scared to fail. Just fail quickly so that you can make the changes that really, really matter. I think this is one that you've probably heard the most, is getting ready for no. You're going to get lots of no's in various different guises. Around fundraising, it's a really challenging environment to fundraise at the moment. There are lots of people that have less of an interest in AMR, but they have it in, they might not have a portfolio that looks in infection control. I think it's just important to remind yourself that doesn't mean that your idea is bad. It just means that you need to iterate or find the talking points that engage those kinds of investors. Just coming back to grants quickly, there were some grants last year, which was brilliant, specifically for AMR, but I think they're few and far between, especially with Innovate UK changing their kind of their funding structure. There's going to be lots and lots more hyper-competitive grants where it's not as simple as submitting something and it's a 50% chance. I think these are down to now 2%, less than 2%. So prepare to have your heart broken in that respect. Trial outcomes, again, that's another thing that's important. You're transitioning something from a lab which has got idealized conditions, and you're going to put it into, from my perspective, real world urines when we were developing our urinary tract infection test. The weird and wonderful is always going to show up in these kinds of environments, but it's important that you do these tests and you find out where you can come a cropper. And similarly with customer feedback. Coming back to the QED example, we're about to go out and show them exactly what we've been working on. So we've got our final box, for lack of a better term. We're readily anticipating that people don't like certain aspects of it. It doesn't mean, again, that the product isn't going to fail. It just means that we need to update or evolve to make sure that this product is successful on the market. So I guess, reinforcing the fact that just like you are in academia, you've got to stay resilient, but you've also got to keep revolving. Don't keep banging that drum without making some important improvements to your processes. And I think what I'd like to finish on, finally, point 5 is collaboration. Talk to everybody in the space. And when you make this first leap into industry, I heartily recommend that you do not do it alone. You don't get any bonus points for suffering independently. So find the help that you need and make sure you ask for it. There are plenty of people on this call. Ask academic founders about their startup journeys. And I think they're all going to tell you the same thing. There's such a steep learning curve when you learn how a business works and how it's different from academia. When you start this new business, you're going to need 1,000 new skills. But I will emphasize, you do not need to be an expert in absolutely everything, but you do need to know how to communicate effectively across your business. So make sure that you understand enough to direct teams, to oversee projects, but you don't have enough hours in the day to do 1,000 things. So again, learn to delegate, but learn to be an expert in your own business as well. But I think just to finish on a positive note as well, the common success story for spinouts, they're those that have a really strong academic founding and they leverage industrial experience to build the right team and then constantly evolve. So, when I say evolve, it doesn't always necessarily mean grow. There are important times where the team needs to shrink, where you need the right set of skills or a different set of skills. And this is where I think consultants can be really valuable. So, because they're always going to offer you the advice you need, even if it's not the answer that you want to hear. So the final lesson really is finding the right people who can help build your venture and taking that next step. So I think that covers, that's me. So I hope our presentation resonates with some of you in the room. And if you think that we can help bring your scientific ideas to life, I'd love to hear about your ideas. So you can contact me directly on my email address or reach out via my LinkedIn. Tell us your problems and let's see if we can find a solution.
Thank you very much, James. And if I could invite Soumya yourself back as well, and we can move on to the Q&A session. We've got about 6 minutes left. Maybe we can take it to 10 minutes if people want to stick around. I've got a few questions coming through the Q&A box, and do please send more if you would like to find out anything now. But I also recommend Soumya and JP that you put your contact details in the chat if people want to contact you. Beyond that, I did that myself. But one of the recurrent questions that's coming up is from people, students really, different stages, undergrad or PhD, asking how they could get more experience in industry and whether there is any internship opportunities available, specifically in the field of antimicrobial resistance. Who would like to take that question? Should I take that to one of you, or I can kick off?
Yeah, do you want to kick off, Magda?
Yeah. So at Cyticon, we actually provide a lot of internship opportunities. I think since I've joined, we've had more than 30 students that we worked with, and in different capacity. It would have been either for a 3-month structured internship where a person was on site working next to me in a sort of funders associate role, really getting to know what the job of the CEO is and helping me with, be it pitch deck generation, market discovery, or other ad hoc tasks. We also had team projects where sort of carrying on the work that I've done as a student when I was working as a management and strategy consultant through societies such as Warwick Consulting Society, or there is also another one called Enactus UK that has a consulting branch. And there we, as a company, worked with teams of about 6 students on a very defined project. There was recently one that we did with finance students who were working on our financial plan and built a financial projection model for Cyticom specifically. But what I found easiest and best when I was a student was to really identify a company that you're quite interested in and you know what they're doing, you like what they're doing, and reach out to them directly. Startups, like JP just mentioned, are always short on resource and they really appreciate the extra pair of hands. And quite often they can say thank you either by paying some sort of internship salary or at least reimbursement for travel and lunch costs. So I would highly recommend that. But don't wait for the adverts to go out on LinkedIn. Just go and reach out.
Yeah, I think I can. I think that's a good point, Magda. I think when you're running a business, you don't look outward that much. So having that inward communication, or people asking, can I help with this, or I saw your paper, I thought that was really interesting. Have you considered this? That's really what gets conversations flowing. And I think in terms of a skill set, if you are ambitious yourself, you're making that first step. You're reaching out to those companies. That's already a positive indicator to suggest, this is someone we can bring onto the team who's going to really help make some progress.
Yeah, I agree. And I think the best is, and this is what we always say to our students as well, if you're really interested in a particular area and you reach out to the company and then ask whether it's on the technical side or on the kind of commercial side, and if you want to get some experience working with the kind of the business development team of the company, the skills that you pick up again is invaluable, and it's good to have. And JP, you were talking about that, that a strong academic foundation. So to have that strong scientific foundation, if you're a student, so understanding, even if you move into the business side of any company, having that sound scientific foundation is really important. Always reach out. We haven't done any, like you, Magda, you were saying, I haven't taken any interns, but sounds like it's a great kind of a scheme to have.
I'm happy to talk to you about that afterwards as well. And one other thing I would add to this as well is that because startups are have got very finite resource, do not get put off if people don't respond to you, or like if we don't respond to you. Just be persistent. Reach out again after a couple of weeks and remind that you've sent a message, because it's unlikely that you are being ignored as such. It's just that the email goes missing or a message just. Some people were too busy at conferences or not working in the office. So do not get put off by that. I'm just looking through the Q&A, but as I'm looking through these questions, another question that I wanted to kind of bring in is, I think it goes hand in hand with what people are interested in, is, are there any specific skills from academia that you brought that you felt really helped you with this journey, and maybe some of the skills that perhaps held you back and you had to grow?
Sumi, do you want to go first this time?
I think so. The main one I would say is the project management skills that so that Magda, I know you said it's different, and I can see it is different in academia versus industry. But for me, because I was heading the Scottish Biologics facility, and we work with collaborators who are also from industry, that kind of bringing project management skills, understanding that the go-to market is or something we all always think about when we work on an antibody discovery program. So the cost of development, the timeline of development, and then managing the project are things that I believe in. I brought in from academia, and it's the same with all academics, PIs who get grants and then they manage a project. So those skills are again something it's really important. Something that so where you wanted to, so you felt like you didn't have much experience and wanted to grow, is that again that the kind of the fit to your product fit in the market or to the customers. So exactly like you said, you have this cool technology, you have this wonderful product, but so what? What is that going to make any change to patients or to other of your customers? So that customer discovery is very important, and I would advise this to anybody who's thinking about this commercialization, that understand your market, validate your market first before you're kind of taking that step. This is something I think in a lot of cases people from academia kind of struggle, because it's your baby, your project, and then but to really understand, is there a need for that product? And sometimes only a simplified version is only needed. You don't really need to go too much into the and don't go again and again and kind of fine tune the product and make it technically more complicated. There's no need for any of that. So understand your customers. Do they really need it, and can you change your technology and make it much attractive to your customers? Because at the end of the day, it's a business; it needs to sell, isn't it? So yeah.
Yeah, if I could just comment. So I think we're all talking very specifically around startups, and I think just to take a step back and sort of give that industry-wide focus. Another piece of advice I give was, industry is a huge catch-all term. Within industry, you've got startups, you've got SMEs, you've got large corporations, and then when you start putting the perspective of AMR, there's human health, veterinary health, environmental. There's lots of different fields you can go into. So I think you're just looking at the kind of general questions that you're asking. What skills do I need? What work experience do I need? I think be more of a renegade when it comes to industry. There's not this formulaic sort of career development that there necessarily is in academia. So if you're looking for an opportunity that fits your skill sets, maybe go, change the perspective slightly. If you're trying to find a job that fits your current skill set, what's to stop you looking for something that's a little bit outside of that Venn diagram, encouraging you to grow a little bit. You've all developed, I presume, fantastic skills practically. Those should be applicable against a huge array of industrial applications. So, don't limit yourselves to being, well, I want to be a research scientist in industry. Have you considered moving into product development? Do you want to manage people? Do you want to manage processes? There's some really interesting fields out there if you look a bit further ahead than opposed to, I am currently a postdoc and I'm looking for a postdoc-like opportunity in GSK or AstraZeneca. Yeah, make sure you just ask yourself the right questions when you're looking for a new job functional role.
I think that's really good advice. And just to close on that, but also add one last thing, is along what you've been saying, James, what I find really important and transformational for me from academia is the networking aspect of it. Mm-hmm. So as an academic, you go to a lot of conferences and you collaborate a lot as well. And really network is your strength. Who you know is what you know and what you can achieve pretty much. So, and you, a lot of you here in the audience, I see as students, what you can do and the advantage you have is to go to a lot of conferences and a lot of events, not just academic, industry conferences as well, investor conferences for free. There is a lot of events out there that will give you free access because you're a student. So capitalize on that, use it, and also reach out to people on LinkedIn, on email, do cold reach out for any advice that you might need. But with that, I would like to close because we're already 5 minutes behind the schedule. Thank you so much again, JP and Soumya, for your time and for sharing the piece of your journey. And thank you to the audience for listening to us. And I will see you next time.
Thanks, everybody.
Thank you. Bye-bye.